welcome everyone. Very nice to have everybody here and a great showing. Thank you. I see 20 participants. So I'm Susan Bacchius and I'm the host of the NVN Reflection Series. This is our first session of the series, conceived as short takes on members' lives. And these are scheduled for the first and fourth Mondays of the month at 7 o'clock. The purpose of the series is twofold: to introduce us to each other in greater depth, and to introduce an opportunity for reflection into our elder lives. The first set of 6 sessions will be vocational reflections, looking back at what for many of us is a completed portion of our lives and giving some thought to how that period, that effort, looks from this point in time, and perhaps how the perspective of time has modified our understanding of the significance of our work. In these interviews, the guest will speak for about 20 minutes. Then, if the guest wishes to, we will take questions for about 10 minutes. Let us begin. I am happy to have as our first guest, Hope Punnett. A few words about Hope. Hope was born in Brooklyn.
Well, actually born in Manhattan, but grew up in Brooklyn.
Okay. And something like 95 years ago, I asked her if I could say that, and she said yes.
I think everyone knows it.
I know it. Okay. She lived around the US and England, but since 1963 has been based mostly in Philadelphia. She's been a member of NVN for about 8 years. She is co-chair of the program committee and serves on the board. Welcome, Hope. And at this point, I'll start it with my question, but you can take this where you want. At this point, how do you remember and evaluate your professional life?
At this point, actually, it's with great sorrow, because I just learned that the most recent replacement in my laboratory has taken another job, and the 2 technicians that are left are scrambling, hoping that the lab will not close. So it's very mixed feelings tonight. So how do I look back on it? I think I was incredibly lucky all the way along, starting out even in Brooklyn.
Excuse me, would everyone mute themselves, please?
When I was growing up in Brooklyn, I was particularly interested in plants and spent a good deal of time at the Brooklyn Botanic Gardens. And at that time, I don't know whether they still do, there was a children's garden. There was someone who was very interested in child education. There were several people who were professional horticulturists. And on Saturday mornings, when we were high school age, we got to hear some of the scientists give their talks. So that was a very wonderful place to start out. Along the way, the 2 women who were horticulturists at the Brooklyn Botanic Gardens both happened to have been Smith graduates. And so that was how I first even heard of Smith. And then I don't know how or why it got into the local newspaper, but there was an article about someone who had retired from directing a very famous laboratory on Long Island, and in his retirement was setting up a genetics group at Smith College. So that kind of clinched it.
Oh, that's
just
great.
Total luck, serendipity, who knows? Good reason to read the daily paper. And at Smith, I had an opportunity to do some experimental work, to meet lots of scientists at Smith and at the other colleges nearby, because Albert Blakesley, who set this program up, started a 4-college genetics group, so that every month or so, a different person would come and speak about whatever it was they were doing. So I got a very nice feeling for what was going on and how academics did science, I guess, and taught. And from there, I went to graduate school, got married, changed my program. I had been doing research at Yale University and Connecticut Experiment Station. We moved to Urbana, Illinois, which again was a matter of great luck, because I had started doing corn genetics, and one of the top corn geneticists in the world had recently moved from Columbia to the University of Illinois. So I've just been lucky all the way along. And when I had a hiatus after that, I had my oldest daughter was born there. We went to Cambridge, England on my husband's postdoc, and my second daughter was born there. We went back to Rochester, New York, and our third daughter was born. So for a period of time, I did not work. Then, this is taking a long way to get to the lab. But again, a stroke of luck. Women at that point could not teach in the same department as their husbands. So early shades of women's lib. There was an opportunity, however, for me to teach night school, because that didn't count as. So I started teaching human genetics at night because nobody had taught it. There were very few books even on human genetics at that point. And there had been a course at University of Rochester, but the man who had taught it had gone to California and had written one of the only 2 textbooks on human genetics. So I started out reading the textbook and teaching. And then a student of my husband's who had gone to medical school said, well, if she's interested in human genetics, the place it's all happening now is at Johns Hopkins. And it just happened that my husband's first sabbatical was going to be in Baltimore. So I did a postdoc at Johns Hopkins. Before that, if I had wanted to do anything beyond teaching at night, the medical school at Rochester was certainly not interested, because I had done my PhD in corn genetics. And that was before I'm blocking. McClintock had gotten the Nobel Prize for genetics, and people began to understand that corn genetics was important. So I was working at night teaching, and then heard this about a laboratory and a department in Baltimore. And in all my innocence, I applied to NIH for a postdoc, little knowing that Victor McCusick, who had started this program at Hopkins, had all the money in the world and could have supported it. It didn't matter. I think I'm the only PhD who was a fellow in the Department of Medicine at Johns Hopkins. And that was where I really began to get some lab experience. And also, because it was really the primary place for human genetics at that time, anybody who was studying any disorder or was interested in anything that had to do with human genetics would come through to meet McCusick, to talk to him, and we would get to hear whatever they were doing. So I got to meet many, many people, some of whom I crossed paths with years later. This is once I finished once my husband's sabbatical ended, and we went back to Rochester, there was someone in the Department of Obstetrics and Gynecology who was interested in human genetics, was doing some studies actually that used human cells in tissue culture. He was not basically interested in the genetics of it, but in placental function. But he had started a laboratory, and I was then invited to come and direct that laboratory. And that was how it all started. We were there in Rochester for 7 years, and then my husband took a position at Temple, and there was a human genetics meeting the summer before we came here, and I met someone named Bill Melman, who was a pediatrician geneticist at CHOP, and who became a wonderful friend and mentor. And Bill said, you have to look at St. Chris. And so I did. And I met all of the great pediatricians who were at St. Christopher's Hospital for Children at the time, and took over a lab that had been started by someone who had been a resident and thought it would be fun to start a lab, and had gone on to pediatric practice elsewhere shortly after I arrived. So that was where it started. And I was lucky enough to just have all of these wonderful pediatricians around me. And as my daughter, who happens actually to be listening to this at this moment, once asked me, what struggles did I have? How did I have to battle? I didn't. I just was very, very fortunate in having all sorts of people help me along the way. And it was wonderful. I'm feeling very sad today because I'm afraid that this lab that I started in 1963 and really was, I think, the second or third cytogenetics lab in the country may be closing because St. Chris is in such a difficult state at the moment, and nobody seems to know where it's going and whether it will continue. But while I was there, all of human genetics was really exploding. We were looking at chromosomes, we were finding out all sorts of things about genetic disorders. I was fortunate in working with other people in pathology and endocrinology, and especially someone named Angelo DeGeorge, who was a brilliant clinician and just a very special person, and was lucky enough to work with him on a study of a patient who really changed the whole way we looked at blood, or at certain blood cells, namely our white cells. At that time, it was not known that there were two different kinds of white cells and they originated differently. And part of the study, which we did with this patient, established the fact that there were T cells and B cells. And somehow I've ridden on that for a long time. It was just, again, great luck to be in the right place at the right time with the right people. Our lab was very active in seeing children with all sorts of birth defects, problems, and there were no genetic counselors at that time. And physicians didn't know very much about genetics, and children with birth defects are all kind of lumped into one category. And so I had to learn I mean, if you do genetics, you have a genotype and a phenotype. The phenotype is the appearance of something. Well, I had to learn about phenotypes in human beings. And once you start making diagnoses and finding out how they're inherited, then you start explaining it to families. And so probably about 4 or 5 years before genetic counseling became an entity into itself, we were doing a lot of genetic counseling at St. Chris. Where to go from there? The lab was recognized nationally. We described a number of different conditions that had not previously been known to be associated with chromosomal abnormalities. And we worked very closely with our hematologist oncologists and did many studies on different forms of childhood leukemia. And then when St. Chris, for a brief period of time, was affiliated with Hahnemann, we did many adult studies in all sorts of malignancies, recognizing that the chromosomal patterns sometimes could be the same in different malignancies, and this would make them suitable for certain therapies. That's kind of a long way around. Something else. What else? We grew cells in tissue culture. Oh, and we also were among the first groups to offer prenatal diagnosis. We would get amniotic fluid, grow the cells, study the chromosomes, and determine. At that point, there were only, in the beginning, there were just a few abnormalities that we recognized that were numerical, because all you could do was stain the chromosomes in the cells, line them up, match them, and see if any were missing or had extra pieces, or sometimes instead of 46, there would be 47, as in Down syndrome, or missing one, as in Turner syndrome. But as different chemical techniques became available and we could differentiate the chromosomes, it became much more interesting in terms of what we understood about genetics, about the way chromosomes interfered with changes in chromosomes could change the way a gene worked. When I started as a geneticist, a gene was something that was immutable. It was, like a string of pearls. And if one pearl came out of place, that was one thing, but you couldn't split a pearl and put it back together with another one. Well, we very quickly learned from the chromosome studies and the biochemistry that went with it, that chromosomes could interrupt a gene, that different pieces of chromosomes could make new genes. The whole world was just exploding genetically at that point. And we were there doing the kinds of studies that helped our clinicians to recognize what was going on. And that became more and more technical. And we learned to do all different kinds of studies of the chromosomes. But then as it moved into the straight DNA studies, it reached the point where I was too old to retool at that point. And it was time for me to retire. So I stayed in touch with the lab. I went in several days a week for a number of years as a consultant. There were patients that came back whom we had seen as babies, who maybe were married now and wanted to know their odds for having children. There were just all sorts of interesting things, and we had all of the records, and most of them I recognized. So it was really just a wonderful feeling to know that you were able to help somebody, even if you couldn't change a diagnosis, being able to tell somebody what caused a problem, whether it was likely to recur or not, how you could test for it, all of those things helped families cope with really disastrous events. When you have a newborn and you're told that there's something dramatically wrong, you know, parents want to know, is it my fault? Is it something I did? We've never had it in the family before. So there are all kinds of questions that you can answer. So part of what I did, even though I was primarily a lab person and I never was qualified, quotes, as a genetic counselor, which today they all are, I had a chance to do a little bit of everything. And that was part of the fun of being at the beginning. Do you have any questions, Susan? I think I've talked for 20 minutes.
Yes, you've done 20 minutes, Hope. Thank you. I feel like I've been on a spaceship and you know, soaring around the universe, a wholly other world where I'm seeing things that I hardly recognize but sort of know. And I've been introduced to so much in a tangential way, but very exciting, Hope.
Well, it was.
Thank you.
It was.
Folks, Hope has agreed to take questions. If you could raise your hand and I'll call on you and you can unmute yourself.
No.
Leonard?
That was really fascinating. But I was hoping that you might tell us a little bit more about your research. I know very little about genetics, but genetic research, but I was really struck by the T cells and B cells. Was that also genetic, that discovery? I mean, that sounds extremely important.
It was, and it's. I'd have to go back. When we first, well, today it's absolutely accepted. I mean, there's no question. Everybody knows there are T cells and B cells. But when this patient turned up at St. Chris, and this is back in the 60s, Dr. DeGeorge we had tried to grow chromosomes because this was a child who was very unusual looking, was a failure to thrive. Nothing seemed to work very well. He had actually already been at CHOP and they had done nothing for him. And so in desperation, the family brought him to St. Chris. And Angelo DeGeorge recognized somehow that he thought that this child was lacking a thymus. And the thymus is a gland that in children is very large and shrinks as we get older. And nobody seemed to know exactly what it did at that time. Well, it turns out that that is one source of lymphocytes. And there is another source which has a different origin in the body. And I'm blocking, I'm sorry. Maybe Joan or somebody could help me. But at any rate, when we tried to grow the cell, the lymphocytes were what we used for looking at chromosomes in those days. We could induce the lymphocytes to divide. And the first times we looked at this youngster's chromosomes, they were normal, but there were cells that divided. But after that, we could never get the cells to divide. So this made us, and especially DeGeorge, think that there was something in the cells, or that this was a form of cell that did not respond. studies were being done in chickens at this time in which two kinds of cells were being differentiated. And when DeGeorge heard about that, it clicked instantly that that must be what was wrong with this child. He also went back and looked at autopsies of other children who had died, who had been lacking a thymus, and we first published a paper on the responsive lymphocytes, and then he published the full diagnostic and clinical and the whole study of this child and other children and the relationship to the two kinds of lymphocytes, which a few very prominent people in the United States immediately jumped on and called it DeGeorge syndrome when it was a constellation of these abnormalities. We now know actually it's due to a very small deletion in a chromosome. So it sort of has gone full circle.
Did that answer your question, Leonard?
Yes, thanks. That was great.
Probably more than you wanted to know.
Not at all.
Any other questions? I can't see who that is. Yes. Barbara? Barbara?
Yeah, that was very interesting, Hope. I'm lagging behind you a bit in the timeline of life. I was in college in 63 and 64, 65, 66. And I had said to studying biology, and the way it was still being taught was like, it was very static. It was like just classifying things and labeling things and remembering things. And then I read a book, Molecular Biology of the Gene by James Watson. And it was like, oh, this stuff is amazing. And suddenly it was, things are moving and things are regulating. And you have your DNA, your protein, your RNA in between, all this stuff. And you were in there at the early times. I mean, these translocations, these DNA rearrangements, you looked at it at the macro level, but as they learn more and more, they realized that you had abnormal proteins. And but it must have just been fascinating that you were in at the ground floor of this. Yeah, and there have been many ground floors since then, with cloning afterwards. But I mean, that book, to my mind, was just, it made biology so dynamic, which is what it should be. It's about life. And it's not just classifying things and remembering them. And it must have been just so fascinating for you to be there.
I think that was what drew me to genetics in the first place, even the little bit that we knew about it.
Yeah.
Well, that and growing plants, because I didn't particularly like the mouse lab or the fruit fly lab. So plants, and also corn has the most beautiful chromosomes to study. So that was another plus.
Myra?
I wanted to add that, Hope, I know you partly as somebody who knows an incredible amount about plants. And I was thinking about you today and wondering whether the Brooklyn Botanic Garden might have been important to you, because I also grew up in Brooklyn and it was an important place to me. And I'm delighted to hear about your experiences, which sound amazing, in the Children's Garden, which they still have. I don't know that it's still that it's as exciting a place as it was when you were there as a teenager, but I know they still have plots for little children and do some education.
I went back, it's probably 20 years now. There was an event in honor of Frances Minor, who was one of the Smith graduates who had influenced me. And it was changed, but it was still there, and I was glad to see it. Yes.
We have a question.
I just want to add, it's not really a question, but just to say, Myra and Hope, Elliot and I grew up in Brooklyn, and we spent many, many, many hours dating in Brooklyn Botanic Gardens. And I have pictures of us in Brooklyn Botanic Gardens.
Hope, where did you grow up in Brooklyn?
I grew up in what the region that the New York Times now calls Madison because of Madison High.
You
Oh,
went to
well,
Madison
Madison,
High?
went to Madison.
And
Myra, how about you?
where did you go to school?
I went to Midwood
And I went to Erasmus.
and Barbara Hoffman.
Oh, okay.
I went to Wingate, which no longer exists.
Oh, my
But
goodness.
my
was- that
parents moved to Madison area, but I didn't want to switch high schools in my senior year.
Well,
But they did
Wingate
move
was,
to the,
Wingate was would have been my regular college, my regular high school. I went to Brooklyn Tech, but I
yeah,
was-
that's because you were a guy and they didn't let
Exactly.
you.
exactly,
That's what was going on back then.
exactly.
Girls weren't allowed.
You're right. You're right.
Or Bronx High School of Science. I
No,
mean,
Brown's High School was co-ed.
Yeah.
That's
Stuyvesant was all
Yeah.
boys also.
Stuyvesant and Brooklyn,
High
which were
school,
much more accessible for me
Yeah.
than
right.
Right.
my mother would never have let me travel to the Bronx. So please, that's
Time for one more question, Robert Harris.
Hope, I know at age 95, it might be tempting to lose touch with all this, but how do you keep up with what's going on? Because I assume you do continue to learn. You're obviously a lifelong learner.
I read things online. I listen to webinars and all sorts of things online. It's amazing how much there is. And I'm still in touch with a number of genetic counselors in the city, so I know a little bit about what's going on in various places also. I don't get into very detailed studies anymore, but I certainly can read a lot of the literature. And I hope you're enjoying Florida, Bob.
Yes.
Had a nice bike ride today. Thank you.
Well, thank you, folks. We're at the end of our time. And I want to thank Hope
Oh,
for coming and sharing her rich and exotic past with us. Next time, our next reflection will be on March 28th at 7 o'clock, and Sarah Allen will be our guest.
Good.
Sarah has conceived a supplement to our interview series, that of capturing our subjects in a sensitive photo. You will have seen the portrait of Hope in the NVN newsletter advertising tonight's interview. That was a portrait by Sarah. Thank you, Sarah, for your contributions. Thank you, Sarah, for kicking off our reflections on our vocations.
thank you. Thank everybody for listening and letting me spout about the wonderful times I've had.
It was great, Hope. Thank you.
Thank you.
Bye.
Good night.
Good night.
Night.
Good job, Sue.